Archons, Mind Parasites, and Extremophiles, oh my. Part 2. Here's a little known fact. Under current law, it's possible to hold a patent on a piece of human DNA, otherwise known as a gene. Some breast cancers are linked with the genes BRCA1 and BRCA2. Companies that have acquired patents for genes have specific rights to their use, which may include diagnostic tests based on those genes, as well as future mutations that are discovered. In a new lawsuit, the American Civil Liberties Union alleges that the policy is unconstitutional. The target of the lawsuit, Myriad Genetics and the University of Utah Research Foundation, hold patents to BRCA1 and BRCA2, the genes responsible for many cases of hereditary breast and ovarian cancers. The U.S. Patent and Trademark Office is also named in the suit. The lawsuit asserts that the patents prevent some people from accessing medical screening for BRCA1 and BRCA2. It also challenges the general patentability of genes, which has been legal since 1980. That year, in Diamond v. Chakrabarty, the Supreme Court found in favor of Ananda Mohan Chakrabarty, who used bacteria to engineer a microbe that dissolves oil. Watch Dr. Gupta explain the lawsuit here. Genes form the basic unit of heredity. With modern technology, researchers have determined that particular genes carry an associated risk of illness. A striking 20% of all human genes have been patented. However, now that all 20,000 to 25,000 human genes have been mapped and sequenced through the Human Genome Project, they are in the public domain, meaning they would no longer be considered new for the purposes of patents, said Lee Silver, professor of molecular biology and public policy at Princeton University. Now, patents on human genes must specify a new use, such as a diagnostic test. If a company wants to patent the purified form of an antibiotic that exists in nature in a fungus, no one challenges that, Silver said. Plant DNA, as well as human DNA, can be synthesized in a laboratory. Distinguishing this case from a patented human gene that is useful in diagnostics would require the ethical argument that the human genome is sacred. And even then, things get murky, considering that about 25% of human genes are shared by chimpanzees, he said. The patent law says nothing about ethics, he said. Again, it is said that power corrupts, and absolute power corrupts absolutely. So, in reference to this notion of power, what would you consider on a scale of 1 to 10, the power base of knowledge in being able to control the entire expression of the human genome within the entire population of Earth, real or synthetic? Would you say that it is a 10 on that scale? Perhaps if one could wipe out the expression for empathy and religious capacity. How about the expression for logical thinking and reason? Would that be a corruption of power? And what about love? What about synthetic people or clones? Who needs the real thing when you can hand-deliver the perfect, docile, subservient beast of a patented human? From its website, the Sanger Institute explains its purpose. What we do. Our research at the Wellcome Trust Sanger Institute builds understanding of gene function in health and disease, as well as creating resources of lasting value to biomedical research. We study diseases that have an impact on health globally by investigating genomes. Building on our past achievements and based on priorities that exploit the unique expertise of our facility of researchers, we will lead global efforts to understand the biology of genomes. We are convinced of the importance of making this research available and accessible for all audiences. Our research into genetics and disease. Our genomic information has a significant impact on our health. Global health problems including cancer, malaria, diabetes, obesity, and infectious disease are partially determined genetically. At the Sanger Institute, we are uniquely placed to build on genome sequences and to engage in biomedical research that elucidates the genetic basis of such common diseases, as well as rare or neglected diseases. Why we study genomes. Genomes are the archival instructions upon which an organism is built. The sequence data provided by the Human Genome Project is a rich source of information that drives improved understanding of human health and variation. Studying human sequences, comparing model organism genomes, and investigating the effects of pathogens on humans will build knowledge of the diversity of our genomes and how this affects our susceptibility to disease. From its annual review, we read, "Genomes continue to revolutionize the study of biology. Their contribution to medicine is just kicking off." In 2011, our sequencing pipeline delivered 200 trillion bases of DNA, more than all the previous years of the Institute combined. Coupled with the output of our genotyping pipeline, which is using the latest DNA chip technology, our researchers are able to interrogate genomes at a scale and resolution that is many orders of magnitude greater than before. This rich vein of information is enabling our researchers to discover and interpret at ever greater depth the significance of genomic variation within and between species. Drawing on our vast genomic resources enables us to play a leading role in national and international collaborations. The techniques, databases, and software we develop allow us to make vital contributions to research into the spread of infectious disease, the development of cancer, and the epidemiology of malaria, sickle cell disease, and metabolic risk factors in Africa. Our ability to answer new questions is turning received genomic understanding on its head. For example, when the Cancer Genome Project team investigated structural DNA rearrangements in the chromosomes in cancer, they uncovered an entirely new mechanism for cancer development. The team discovered that some people's cancer genomes bore the scars of a catastrophic event that had driven them a number of steps towards cancer in a single-cell cycle. The chromosomes had exploded, and incorrect pasting together of the DNA had produced many cancer-driving mutations. Another convention-busting discovery was made when our researchers sought to complete the genomic picture of the spectrum of Leishmania parasites. Seeking to understand the genomic variations responsible for the differing severity of symptoms caused by the different strains, the team made a surprising discovery. Producing high-quality reference genome sequences showed that the different strains had almost identical genetic codes, but that the strains' genomes contained different numbers of copies of the chromosomes. This finding suggests that the parasite's evolutionary development and success is founded on the numbers of particular genes and chromosomes it has. A genetic abnormality that would kill most organisms. Our pathogens team are harnessing our high-throughput sequencing and analysis resources to compare variation between individual bacterial and viral genomes from patents to track and trace precisely the spread of disease. By comparing cholera genomes during the current global pandemic, our researchers have been able to categorically trace its origin back to the Bay of Bengal. Using the same technique, we have mapped the spread of the H1N1 flu virus across the UK during the most recent epidemic, and have shown that the disease entered at a number of geographic locations at different times, before the first clinical case was identified. Our ability to conduct genomic research at high resolution and vast scope enables us to provide the foundations for the global research community to build upon. For example, the fruits of our investment in the 1000 Genomes Project are being harvested by researchers across the globe as they use the data to enhance the resolution of the genome-wide association studies and understand the origins of genetic mutation. Open source genetic codes for genomes? Instructional genomic blueprints for cancer offered to the world. Offering the entire mechanism for the spread of disease on the cellular level to the militaries, governments of the world. Sharing the origins of genetic mutation with anyone interested. 20 trillion base DNA sets deconstructed in a single year. Perhaps my concern is not being completely burned into the reader's consciousness here. And perhaps that's because the reader may not be reading this from the point of view of a racially driven, psychopathic purist depopulationist. Get it? The Sanger Institute? In honor of dear old Margaret? This should make even the roots of your own DNA shake violently with fear and loathing. Satan, if you believe in that sort of thing, could not ask for a better tool as the defined adversary of man, with this tool set being the epitome of the model chemistry set to deconstruct nature and reform it in man's own image. This new world order of DNA and genomic expression belongs in no man's hands, for no man is immune from the ultimate disease of unrestrained power. There are no true ethics in the modern notion of what is science, because the institution of science today specifically disregards nature's, God's, design in order to exploit and alter the intent of that self-evident design. It studies nature only in order to change or destroy it. The diseases it is claiming to be able to treat and cure on the genetic level were, of course, caused by this very same institution of medical and scientific crimes against man and nature in the first place. And so the placing of our faith in such institutions to cure us from its own self-inflicted ills is foolhardy at best. Cancer was all but non-existent before modern practices of those who profess to be professors, professionals, scientists, and doctors spread it experimentally as vaccine ingredients, rearing its ugly head, according to some in history, only after modern vaccination was introduced. And that brings me to my point. We have all been stung by this beast of medical science. Vaccination is designed to apparently replace what nature has already installed within the expressions of the body as the natural immune response to outside influences. One has no allergy to bee stings, for instance, unless one is stung by the bee, which delivers its disease-causing agent through a stinging injection. Spiders penetrate by a stinging bite, as do dogs with rabies. Tetanus and hepatitis thrive many places, but can only enter the body through a stinging penetration of the skin, or through the penetration of intercourse, i.e. the exchange of bodily fluids. The vaccination needle is the simulated, weaponized sexual appendage of the science of modern medicine. We are violated by it, raped by it. We are injected, stung, with substances and DNA that would never otherwise have the capacity to enter our bodies and mix with our substance. We are literally being grafted like fruit trees with the foundational building blocks of the gene expression and DNA of other life forms, including that of our fellow man, as with the injection of cloned human diploid cells, including DNA and proteins, from various aborted fetal tissues, human albumin, from blood, rhesus monkey fetal lung cells, continuous, cloned line of monkey kidney cells, rhesus monkey fetal diploid cells, simian cancer virus 40, and at least 79 others, vesicle fluid from calf skin, calf serum, bovine fetal serum, U.S.-sourced bovine extract, washed sheep red blood cells, RBCs, chick embryo, chick embryonic fluid, chicken protein, mouse serum proteins, guinea pig embryo cells, shark squalene, gelatin, hydrolyzed gelatin, processed gelatin, lactose, and others. These are just a few of the ingredients that you have been infected with by stingers called needles. Vaccination is, in the most simple description, the purposeful infection of the body with foreign particles and substances. Whether those substances are good or bad, beneficial or harmful, therapeutic or deadly does not remove the fact that vaccination is nothing more than purposeful infection with disease. This is not contagion, for a vaccinated person is not necessarily contagious, though some shedding of the vaccine does take place after infection, vaccination. The injection of peanut oils and lactose as ingredients in vaccines, for instance, is certainly linked to localized milk and peanut allergies that are not spreadable as contagious infection to others, but are rather local reactions to otherwise harmless foods, if only they were eaten instead of injected past any natural barriers. Thus, we must think outside of the box we are placed into by media education and realize that infection is a neutral word that actually also represents any forcing of so-called medicine into the body. But consider how the body then might react to monkey kidney tissue or cow blood being injected into it, bypassing the natural protective barriers for such agents through the deep penetrating sting of the inoculation needle. It is well known that even the Rh factor of some human blood types prevents negative and positive bloods from mixing to create life. The mother's body will literally attack the newly formed embryo of a different blood type, Rh, to kill it as a foreign infection. So imagine how incompatible your body and its blood and fluid is to cow, monkey, pig, sheep, insect, and other animal blood and protein products used in vaccines, which have no other way to enter and infect your body but through the penetrating sting of vaccination. But let's not stop there. For other ingredients within vaccines also have no hope of forcibly entering past your body's defenses without the sting of a nurse or pharmacy technician with a couple of weeks of government-sponsored training and indoctrination mixed with a healthy dose of cognitive dissonance. The arrogant advocate for vaccination is always one whose livelihood depends on delivering it, no differently than those animals and insects that sting or bite in protection of their own livelihood, or to spread parasites. And the propaganda machine of fallacy and quack science fills the heads of those who subject themselves and their own children to such violations of the natural law as vaccines are. Let's not forget the heavy metals and other extra ingredients also used in vaccines, with thinly veiled reasonings and names such as preservatives and adjuvants. Those include such other foreign particles and poisons, medicines, as formaldehyde, aluminum hydroxide, aluminum phosphate, thimerosal, mercury-based, polysorbate 80, tween 80, ammonium sulfate, formalin, sucrose, sorbitol, benzethonium chloride, glycerin, phenol, a compound obtained by distillation of coal tar, beta-propionolactone, 2-phenooxyethanol, polysorbate 20, yeast, chemically defined yeast-based medium, soy protein, phenyl red indicator, phosphate buffered saline, monosodium L-glutamate, MSG, potassium glutamate, potassium chloride, potassium phosphate monobasic, potassium phosphate dibasic, potassium monophosphate, potassium diphosphate, sodium bicarbonate, sodium phosphate dibasic, polydimethylsiloxane, silicone, and even the known cancer-causing agent aspartame. It is therefore likely that in yours, and especially in your child's lifetime, most or all of these listed vaccine ingredients have been injected into your body, along with the various disease-causing microbes, antibiotics, anti-life, and other ingredients not listed here. And your body reacts to every one of these ingredients, either in the short or long term, as the manifestation of chronic disease states. I can think of nothing else to call this but sheer insanity. The blanket acceptance of the science of vaccination in the mind can only be attributed to two things. Ignorance is certainly a fine-tuned and well-oiled machine within the general population. But can we really blame sheer ignorance? What about custom, routine, greed, profits, and medical advice? But what if there is another factor at play here? What if there is another unseen force that is driving people to promote and commit to actions like vaccination, eating junk food as their main staple, taking pharmaceutical drugs that will knowingly cause more severe disease than what they will treat, and submitting themselves to the unspeakable atrocities of modern and cosmetic medicine, which is, by the way, iatrogenic death, or death by medicine or doctor? Is it possible that mankind could be driven subconsciously by another infective force, more powerful than ignorance, more persuasive than propaganda, and more controlling than hypnosis? And if this possibility may indeed be the case, would man even know he was infected? This is the theory I'd like to explore herein.